Hello class and welcome to our second video. We are still continuing on with the topic of DNA and proteins, and this video will be on DNA replication. So as always, we have our science understanding at the beginning of the video, using this as a checklist so that by the end of the video we should be able to tick off each of these in accordance with our subject outline. So replication of DNA allows for genetic information to be inherited. Base pairing rules and method of DNA replication are universal, whether that be prokaryote or eukaryote. Describe the structural properties of the DNA molecule, which we should be able to do already. This includes the nucleotide composition and pairing, and the weak bonds between the strands. This is explaining the importance of complementary base pairing, as well as describing and representing the process of semi-conservative replication of DNA. Let's get into it. So an overview. We already know that DNA contains all our genetic information. This must come from our parents. So every single cell that is made in your body, that has come from a pre-existing cell. These are our parent cells, and the new cells being made are our daughter cells, and they are genetically identical. So let's go over these rules yet again. We have our DNA molecule composed of two polynucleotide strands. So a nucleotide is just one section of this DNA. We have our sugar and our phosphate, making our sugar-phosphate backbones, and our bases, our nitrogenous bases, which pair in accordance with each other. So we can only have our adenine to our thymine, or vice versa, and our guanine to our cytosine. This is known as complementary pairing, and each sequence must mirror with its counterparts. This allows for two identical copies of DNA molecule in a cell preparing to divide. Let's get into it. So, before a cell divides, it must possess one or more chromosome that needs to be copied. In humans, we know we have multiple pairs of chromosomes. We should know that number is 23. The daughter cell must have a complete set. If it does not have a complete set of the DNA, the cell will not function, as the DNA is faulty. And so the process by which chromosomes are copied is called DNA replication and is the highlight of this video. A chemical reaction separates the parent DNA so that free nucleotides can join on and construct a complementary strand. The daughter DNA is then made, ready for cellular division. So, if this is our parent strand, in the blue, in the green we have our free nucleotides joining on to our bases to form that counterpart. Now there's going to be multiple steps within this video that show this replication in more depth each time with enzymes that are present. So, consider this the simplified version of DNA replication, and this is the one you must know. If you know anything less than this, you do not understand or know of replication. So first, DNA strands are separated by an enzyme called helicase. This unzips the DNA, so we get two strands on either side, and this makes our fork-like structure. It breaks the weak hydrogen bonds between the bases so that they can separate. Next, we have our DNA polymerase. DNA polymerase is responsible for binding these free nucleotides to the new strands. Okay. Once it has bound these nucleotides, it also has the sugar phosphate backbone, which is bound to it as well. And that, in a nutshell, is the replication of DNA. So, there will be more and more details to this, and if you go off to do your own research, you might get lost in all the other components that are present within DNA replication. Please feel free to ask me in class. So, we can see our parent DNA. We know that DNA runs 5' to 3', and it's important in replication as to how the DNA polymerase will function. The helicase unzips these bonds, and we notice that our polymerase, our enzyme, is attaching those free nucleotides to form the new strand. Okay. Oh, I said okay. Was that my first one? Okay. Moving on. Let's have a look at it in a bit more depth. So the way DNA polymerase can work is it can only add those nucleotides in the 5' to 3' direction. So if we have our original strand that's unzipped, let's have a look at this 5' one. We see that this end of the DNA is considered 3'. So the polymerase can attach here and it'll add the DNA from the 5' to the 3'. In our strand that it is going in this direction, it is known as continuous, and this is the leading strand. It's very convenient for the DNA. The polymerase can just keep on adding those nucleotides as the DNA unzips. It gets this continuous feed. However, in the other strand, where we now have our original strand acting as 5' and the newer strand being 3' on this end, the polymerase must go in the opposite direction to add its complementary strands. However, by doing this, it is going down the DNA strand, okay? Which means as it goes down, you're not getting this newly fed strand. So the DNA polymerase has to jump back each time to keep on getting the new strand. This is called the lagging strand. And so what it does is it makes this DNA in different fragments known as Okazaki fragments. And these still run 5' to 3'. So this is 5' on the lagging strand, running down to 3'. So that on our new strand, the 5' would be here and the 3' would be going in this direction. So the leading strand is continuous, the lagging strand is discontinuous. And so what we can say is this DNA polymerase has to sort of catch up in fragments. That's how I like to picture it. It's in a constant race to get back to where the new DNA is so it can keep adding the nucleotides. Okay. It's getting a little more complex now. The key components to take away from this is we still have our helicase that is unzipping our DNA. We have our leading strand at the top, as the 5' will be running from this direction. So 5' will be here, 3' will be down here. On the opposite side of the strand, it should be mirrored. So now we have our 5' up here, going all the way down to our 3' down here. Okay. Some other things we can see in this picture is we have our DNA polymerase. This is adding our nucleotides. But now we have something else known as a RNA primer or RNA primase. The primase is responsible for starting where the DNA polymerase will go. So it primes where it should start. It acts as a checkpoint for the DNA polymerase. It goes, this is the primase, I need to go here and keep on making nucleotides. There is only one primer present on the leading strand because once that DNA polymerase has attached, it can continuously add those base nucleotides. However, in our lagging strand, we have to keep on adding these primers so that the DNA polymerase can catch up. We get our Okazaki fragments from where the DNA polymerase has made a fraction of the daughter strand. It must then catch up to where the new primer will be. These are our nucleotides just being added. And we also have something called DNA ligase. Think of this as the glue. I'm not dying yet. The ligase, where there are gaps in the DNA, will fill them so they are connected as a full strand. Okay, there's lots present within that. So a question for you is, why would DNA replication be called semi-conservative? I'll give you 5 seconds to have a think about that before I go over why. So what we have is our picture of our DNA replication. In the blue, we have our parent strand. In the red, we have our new strand being added so we can form our daughter DNA. The reason it is semi-conservative is for every process of DNA replication, those two cells that you are making, one will be your parent strand and one would be your daughter strand. So the reason it is semi-conservative is it will always have the genetic information from one of the original strands. We've discussed how in eukaryotes DNA is significantly longer compared to prokaryotes, and due to that, DNA does not just have this one site of replication, it can have multiple sites. And so what we get are these little bubbles where we get replication forks. The DNA replication will start, and as it begins to expand and run both ways, these start to join up so that by the end of it, you have your two parent strands. Okay, just quickly on inheritance. So this genetic information is passed from parent to daughter cells. The prokaryotes inherit this through a process called binary fission, which we will talk about more in later videos. And in the eukaryotes, we inherit it through mitosis and meiosis. So the image on the right is showing a full cell replication cycle. The only section we are looking at today is the S phase. So this S phase is where the DNA replicates. We'll break down all these other stages later, and we'll even look at what these checkpoints are. But for today, we are just looking at DNA replication. It takes place in the synthesis phase, hence why it is called the S phase. So before the DNA is replicated in eukaryotes, and this is two or more linear bodies, they are called unduplicated or unreplicated chromosomes. Once they have replicated, who would have thought, they are called replicated chromosomes. These replicated chromosomes are connected to form daughter DNA molecules. They are then pulled apart during cellular division. But replication, again, only occurs in the S phase. So why would replication be faster in prokaryotes compared to the eukaryotes? Again, I'll give you 5 seconds. Okay. Oh, I said okay again. I'm aware of that one. All right, we have our linear DNA in our eukaryotes. These are our replication forks for our new strands being made, and these are origins of replication. In our prokaryote, we have our circular DNA. We have one replication bubble, so it only starts at one point, and our replication forks that will run opposite directions. What was one of the biggest differences, other than the shape, between the eukaryote and the prokaryotic DNA? Hopefully, in our minds, we're thinking about the size. Because the prokaryotic DNA is typically significantly smaller, we should expect that this replication time can be a lot quicker. And that's it. That's DNA replication. So hopefully, we know that replication allows for this genetic information to be inherited. The base pairing rules we are very familiar with. We know why there are weak bonds, so they can be broken by this helicase. And we can explain the importance of complementary base pairing. If it wasn't complementary base pairing, we would not get these perfect daughter strands. And we know why it is called semi-conservative, because one strand will be from the parent and one is the newly formed daughter strand, giving us two new daughter DNA strands. Thank you. We'll talk about this more in class, and we can unpack more of what this replication is if you have done additional research. It is a very in-depth process, so if you are looking into this outside of these resources, please note that you might get lost. Try and strictly stick to the things that are in this video. Thank you.